Hello students, welcome to the EPG Pathshala. I am Dr. Sarita Kumar, working as Associate Professor in Zoology at Acharya Narendra Dev College, University of Delhi. Today I am going to talk about module, Morphology, Life Cycle and Transmission of Wuchereria bancrofti from the paper Parasitology. Have you ever heard of this organism? To which phylum it belongs to? Wuchereria bancrofti is a thread-like cylindrical nematode. It belongs to phylum Nematoda and class Phasmidia. You have learnt about nematodes earlier. You must have listened about or you've heard about Ascaris. That is also a nematode. Nematodes are pseudocoelomates, non-chordates, whose body cavity is not bounded by mesodermal lining. Their body is covered with a thick cuticle with syncytial or you can say multinucleated epidermis. Majority of them are parasites, but a few are found as free living forms too in soil, etc. Today we will try to realize the medical importance of this parasitic nematode, Wuchereria bancrofti. Identify the parasite from its morphological features. Distinguish between male and female Wuchereria bancrofti. Recognize its genetic nature. Comprehend its life cycle in two different hosts, and finally understand the factors affecting its transmission in human beings. Before going into details, let us first explore the features of Wuchereria bancrofti. Wuchereria bancrofti, also known as Filaria bancrofti, is a parasitic nematode of human beings. It is commonly known as human filarial worm and is well known as the causative worm of a very peculiar disease called lymphatic or Bancroftian filariasis, or you can say commonly elephantiasis. You may have come across the individuals suffering from this disease. In this disease, the lymphatic and genital organs enlarge because of lymph accumulation. The organs enlarge so much that they look like as the organs of elephant. These organs get disabled either temporarily or permanently, and the human being is unable to move. The disease is extremely painful and disfiguring kind of disease. The infection of this disease is generally attained during childhood, but often visible manifestations occur later in life. It may take many years to be visible. The parasite Wuchereria bancrofti is named after its two discoverers. First, Brazilian physician Otto Edward Henry Wucherer, who discovered the larvae of Wuchereria, and next one is British parasitologist Joseph Bancroft, who suggested its transmission by mosquitoes. However, the parasitic effects of Wuchereria bancrofti have been acknowledged early in prehistoric texts. If you go through literature, you can find the description of a filariasis-like disease has been documented in 6th century BC. You can read that description in chapter 12 of the Susruta Samhita. Signs and symptoms of this disease are also described in 7th century AD by Madhavakara in chapter 39 of his work Madhava Nidhana. Do you know what are the signs and symptoms of this disease? In 1709, Clark named elephantoid legs as Malabar legs in Cochin. That is the sign and symptom of this disease. Whereas Microfilariae were discovered for the first time in peripheral blood by Lewis in 1872 in Kolkata. Where can you find this parasite geographically? Is it present everywhere throughout the world, or it is confined to some portions only? Yeah, the worm is largely confined to the tropical and subtropical regions of the world only. It is known to affect only 73 countries, which include India, West Indies, South America, West and Central Africa, Southern China, Japan, Pacific Islands, and Korea. You may be surprised to know that approximately 120 million people are infested with the disease filariasis, out of which about 25 million men suffer from genital disease, while 15 million, mostly women, with leg elephantiasis. Not only this, but approximately 66% of those at risk of infection live in the Southeast Asia region, and 33% people live in the African region. Interestingly, the disease is completely absent from Europe, North America, and polar regions. In United States of America, the infection had disappeared early in the 20th century. There, only 4 countries are currently known to be endemic: Brazil, Haiti, Guyana, and Dominican Republic. You may be wondering now, what about India, where you can find Wuchereria bancrofti in India? In India, Wuchereria bancrofti is primarily distributed in the southern regions only, especially along the sea coast. It is known to affect more than 100 million people in India itself. Native cases of the disease filariasis have been reported from about 250 districts in 20 states and union territories. Major cases have been recorded from Pondicherry, Andaman and Nicobar Islands, Lakshadweep, Daman and Diu, Dadra and Nagar Haveli, and Goa. However, on the other hand, northern eastern states are known to be completely free from indigenously acquired filarial infection. Now, what is the mode of transmission of Wuchereria bancrofti? How does it cause the disease in human beings? Before going into details of the life cycle and pathogenicity and transmission of Wuchereria bancrofti, let us first try to understand about the distinguishing features of the earthworm and its habit and habitat. As we have discussed earlier, Wuchereria bancrofti is a thread-like cylindrical nematode. It is endoparasite of only human beings. It is not found in any other mammal or any other animal of any species which can act as primary host of this parasite. In human beings, it is present in the lymphatic vessels and lymph nodes of the body. As you can see in the figure, there are so many lymph nodes present in the body, and lymphatic channels are distributed throughout the body. However, it is present particularly in the groin regions where it causes the disease filariasis, but it may be found in other lymph nodes and lymphatic vessels of the body. Now the question arises, how and where do these parasites multiply? Wuchereria bancrofti is a digenetic parasite, unlike other nematodes, or you can say Ascaris lumbricoides, which is a monogenetic parasite. Wuchereria needs 2 hosts to complete its life cycle. The definitive of primary host are specifically human beings. That means the sexual part of the life cycle of this parasite can take place only in human beings. It cannot multiply in any other mammal of any species. The intermediate or secondary host where asexual part of the life cycle occurs can be various species of mosquitoes. You may have heard of many species of mosquitoes, for example, Culex, Aedes, and Anopheles. The Wuchereria bancrofti can reproduce in all 3 species. However, in India, only Culex pipiens fatigans serves as the secondary host. In Pacific Islands, Aedes polynesiensis occurs as serves as the secondary host of Wuchereria. There are no reservoirs for this parasite. Now you may be thinking, where does the parasite live in man? Adult Wuchereria bancrofti resides in the lymphatic system of the human host. Male and female both live together, coiled with each other so strongly that it becomes difficult to separate them at all. Copulation between male and female adult takes place in the lymph glands of human beings. During copulation, male adults of Wuchereria bancrofti grasp females with the help of coiled tail region. As you have learned earlier, that males have anal papillae and copulatory spicules around the cloacal region. These spicules help them to open the vulva and vagina of females and transfer the sperms. Scientists have proved the involvement of sex pheromones in attracting mates. In addition, evidences suggest that males possess a chemosensory apparatus required for recognition of such signals. Now, how does Wuchereria bancrofti reproduce within the human beings? Do you know that? The females of Wuchereria bancrofti are ovoviviparous. You may have heard of ovoviviparous. What does it mean? Instead of laying eggs like many other nematodes, they liberate numerous active embryos called juveniles or microfilariae in the lymphatic channels of human beings. It means they do not lay eggs, but they directly release juveniles in the lymphatic system. Microfilariae are elongated and filiform larvae with blunt head and pointed tail. They are approximately 200 to 300 micrometers long and 6 to 7 micrometers wide. If you observe the figure of Microfilariae carefully, you can see these features very clearly. Now you can see the male Wuchereria and female Wuchereria differ from each other in many features. The shape of male Wuchereria is smaller and thinner than the female adults. Females are much longer and stouter than the male adults. Male Wuchereria are 2.5 to 4 cm long, whereas the length of female Wuchereria ranges between 5 to 10 cm. Similarly, the males are 100 micrometers broad, whereas the width of female Wuchereria is between 200 to 300 micrometers approximately. Posterior end of male Wuchereria is sharply curved ventrally, whereas the posterior end of females is much narrower, which tapers gradually and has the rounded tip. Male Wuchereria has a single opening called cloaca. The cloaca is a common opening of digestive and reproductive system, whereas females have two openings instead of one. Their digestive system opens into anus, while reproductive system opens into gonopore. The cloaca of male Wuchereria is present at the posterior end, whereas the gonopore of female Wuchereria opens ventrally at the anterior 1/3 of the body. Male Wuchereria have pineal spicules, genital papillae, and sensory papillae, which are completely absent in females. In males, there are genital papillae which are present at the anal end, whereas sensory papillae are total 15 pairs, which are present at the again anal end. The 11 pairs of sensory papillae are present before anus and they are called pre-anal papillae, whereas 4 pairs are present after anus and therefore they are called post-anal papillae. Now what happens to these larvae? Do they multiply humans? How and where do they develop? The Microfilariae are very active larvae. When adult Wuchereria bancrofti release them into the lymph vessels, they quickly enter the main lymphatic trunks. Eventually, they find their way into the circulating blood and they start moving through the blood into different organs of the human beings. Though generally they move along with the blood streams, but they are capable of moving against the bloodstream too. Ultimately, Microfilariae migrate to deeper blood vessels and stay there for further development. These blood vessels can be in various organs of the human beings like kidney, liver, etc. Microfilaria bancrofti, the larvae of Wuchereria bancrofti, are elongated, filiform with blunt head and pointed tail, as you can see in the figure. Their length is only 200 to 300 micrometers approximately, and the width is only 6 to 7 micrometers. As you have learned earlier that the length of Wuchereria bancrofti is around 4 to 5 centimeters. So you can see that how much growth they have to attain. Microfilaria bancrofti are colorless, or you can say they are transparent. You can't see without staining. They are covered with a hyaline or transparent sheath. As you just now learned, that body of Microfilaria is covered by a transparent hyaline sheath. The sheath is 359 micrometers long, and you can see in the figure that it is much larger than the body. Why is it so? The ends of the sheath project beyond the ends of the embryo so that embryo can move easily within it. Further, the cuticula of the body wall is lined by specialized subcuticular cells. Another diagnostic feature of Microfilaria is the presence of numerous nuclei in their cytoplasm, just like the nuclei are present in the adults. These nuclei, as you can see in the figure, are present as granules, but they can be seen clearly only after staining. Otherwise, these granules are not visible at all. However, as you can see in the figure, You can see that certain areas of Microfilariae lack these nuclei. These absence of these nuclei serve as another distinguishing feature of Microfilaria. These areas signify the anterior end called cephalic space and tail end called anal space. Further, if you see the figure carefully again, you will observe that granules are broken at definite places, which help in the identification of species. These places are those which will later develop into nerve ring, excretory system and anus or cloaca, depending upon whether the Microfilaria will develop into male or female Wuchereria bancrofti. The body of Wuchereria bancrofti adult is hair-like and transparent with tapering ends. You can see in the figure clearly dark granules dispersed throughout their body cavity except in the tail end. These granules are actually nuclei. They have a small mouth devoid of buccal capsule. However, the most characteristic feature you can observe is the slightly enlarged head terminating in a slightly round swelling. The head also has two circlets of distinct papillae and is connected to the main body by a short and constricted neck. Wuchereria bancrofti is a sexually dimorphic species. Males and female are separate and differ from each other morphologically. Adult females are much longer and wider than the males, the females being 5 to 10 cm long and 0.2 to 0.3 cm in diameter, whereas male is 2.5 to 4 cm in length and 0.1 cm in diameter. Like other nematodes, the gonopore and anus are separate in females, while males have a common opening called cloaca. A pair of copulatory spicules also emerge from this opening. Further, you can see that male adults have a ventrally curved tail. It has 15 pairs of minute caudal papillae at its tip, which are sensory in nature. Out of this, 11 pairs of papillae are present in front of anus and are called pre-anal papillae, while 4 pairs are located behind the anus and are named as post-anal papillae. On the other hand, the tail end of female worms gradually tapers, is rounded at the tip and lack any papillae. There are no additional sensory structures present in both of them. Let us summarize at these features again. As you can see from the figure, the circulation of Microfilaria occurs within the lymphatic system of the body. Earlier they are present in the lymph vessel, then they pass on to lymphatic trunk, then they are present in the bloodstream, or you can say they move to the bloodstream. The average lifespan of Microfilariae in human body is approximately 70 days. However, they cannot develop further as they need a lower temperature for their development. Therefore, they stay in human beings as newly emerged larvae unless they are sucked by their intermediate host. As we studied earlier, that in India, Culex pipiens fatigans is the principal intermediate host. If they are not sucked by these mosquitoes, they die and disintegrate in the human body. You may be aware that Culex is a nocturnal feeder. It feeds only during night because it needs darkness to feed upon human beings. Thus, Microfilariae have developed a specialized system to be sucked by these mosquitoes during night. During daytime, the Microfilariae reside in the large and deeper blood vessels of different organs of human body such as lungs, kidneys, heart and large arteries, whereas during night they appear in the peripheral blood vessels, especially between 10 p.m. to 4 a.m. so that mosquitoes can suck them easily. This is called nocturnal periodicity. As you have studied earlier, that in Pacific Islands, Aedes polyneesiensis is the intermediate host of Wuchereria bancrofti. You may be aware that Aedes can feed on human blood throughout the day and night, unlike Culex. Therefore, in those regions, the Microfilariae do not exhibit any periodicity and they are found in the peripheral blood throughout the day. You must note here that only female mosquitoes can suck the blood. Male mosquitoes do not need blood as blood is required for the maturation of eggs laid by the female mosquitoes. Now the question arises, how does Wuchereria recognize its environment? How does it know that it has to come in the peripheral blood to be sucked by the mosquitoes? It has been reported by various scientists that the worm lacks visual abilities and depends on its sensory receptors and tactile papillae to detect chemicals in its environment and pheromones released by other members of its species. However, the periodicity of the worm is often correlated with the lifestyle patterns of its human host. The worm detects the difference in oxygen levels between arterial and venous blood vessels. This difference is indicative of decreased oxygen intake and lessened host activity during night time, that is, the feeding time of Culex mosquitoes. Thus, when Wuchereria bancrofti senses even a small difference in the oxygen content between venous and arterial blood vessels, it migrates to the peripheral circulation. It increases the chances of ingestion by Culex mosquitoes, leading to dispersal to other hosts. Now, when female Culex adults suck the blood of an infected individual, the sheathed Microfilariae are also ingested with the blood. They reach the stomach of mosquito where they lose their transparent sheath. Within 1 to 2 hours, the Microfilariae, which are now without any sheath, penetrate the wall of stomach of mosquitoes and migrate to their muscles of thorax or wings. Further development of Microfilaria now takes place in mosquito, where they pass through different larval stages, where there are 3 larval stages in the mosquitoes. It takes approximately 2 weeks. First stage larva develops in the next 2 days of their migration to the thoracic muscles. It measures only 124 to 250 micrometers in length, 10 to 17 micrometers in breadth, and is much thicker, shorter, and sausage shaped with spiky tail. Within next 3 to 7 days, the first stage larva sheds its cuticle. It becomes thick, enlarges, and metamorphoses into second stage larva. Now it measures approximately 225 to 330 micrometers in length and 15 to 30 micrometers in breadth. The second stage larva enters into third stage on 10th to 11th day of development. This is almost 5 times in size to that of second stage and measures about 1500 to 2000 micrometers in length and 18 to 23 micrometers in width. The spiky tail degenerates while various organs such as digestive and genital organs and the body cavity are now well developed. On about 14th day, the third stage larva migrates to the proboscis of the mosquito. As you can see in the figure, how mosquitoes suck the Microfilariae from the human body, which pass through their gut and muscles and develop into third stage larva, which finally reach to the proboscis of Culex female adults. The Microfilariae now cannot develop further in the mosquitoes as they need higher temperature for maturation into adult Wuchereria bancrofti. Therefore, the third stage larva waits for the new human host for its development. You must note here that the third stage larva is infective stage of the filariasis disease. The infection now takes place to a new human host when the infected female Culex mosquito sucks the blood of a healthy human being. Unlike other parasites, while sucking blood, the mosquitoes do not release the third stage larva in human body. You may be aware that in case of malaria and other diseases, mosquito release parasites such as Plasmodium directly into the human blood. However, in case of filariasis, the microfilaria are not released directly into the blood. Instead, these are released on the skin of the host near the site of puncture. The larvae get attracted by the warmth of human body and they invade the skin either through the puncture or on their own. They migrate through the subcutaneous tissues, reach the lymphatic vessels, and accumulate at a particular lymphatic region. The larvae accumulated in the lymphatics now go through two molds and begin to grow into mature. It takes them approximately 5 to 18 months to gain sexual maturity. As we discussed earlier, the male and female started living together in coiled forms and undergo copulation. Female Wuchereria bancrofti now gives birth to new generation of Microfilariae and whole cycle repeats. You can watch whole life cycle in the given link. As you can see from the figure, the complete life cycle of Wuchereria bancrofti occurs in two phases. One, human phase, where they undergo sexual reproduction and release the microfilaria into the blood. When these microfilariae are sucked by the mosquitoes, they develop into third stage larvae within the mosquito, and now when they bite fresh human host, they release third stage larvae into the human body. Therefore, as sexual life cycle of Wuchereria takes place in the human body, the human beings are called primary host, and as asexual part of the life cycle, or you can say development of the larva, takes place in the mosquitoes, mosquitoes are considered secondary host of the Wuchereria bancrofti. Do you know that the transmission of Microfilaria bancrofti is considered to be less efficient than that of other vector-borne parasites such as malaria parasite and dengue virus? Why so? Can you guess? This is because of various factors that limit the filarial worm. For example, the transmission depends upon the number of microfilariae ingested by mosquitoes, as the microfilariae do not multiply in the mosquito body. They just develop and are then transmitted to a new human host. Similarly, the transmission depends upon the longevity of mosquito. You studied that the development of microfilariae in the body of a mosquito takes about 2 weeks. Thus, only those mosquito adults that have more than 15 to 20 days of longevity will be able to contribute to transmission of the parasites. Further, as the mosquito does not inject third larval stage into the human body but deposit them on the skin near the puncture, the larvae may not enter the human body until they find their way into the bite wound. Along with all these factors, the transmission of parasites also depends upon the biting rate of mosquitoes, number of infectious hosts available from which they can suck the blood, its possible adverse impact of mosquitoes, and the local environmental conditions such as rainfall, temperature, humidity, and soil type, which can affect the breeding sites of the mosquitoes and the survival of them. Thus, in order to disrupt filarial worm transmission and to ensure that no new infection occurs, the intensity of microfilariae or the vector density must be brought down below a threshold. It varies because of the heterogeneity of the vector-parasite relationship, which can be evaluated by various indices. These indices include transmission potential of a mosquito, means how much a mosquito vector can transmit the disease, its longevity, etc. Similarly, percentages of mosquitoes infected with filarial worms, and especially those mosquitoes infected with third stage larvae, also decide the prevalence of disease, because when a mosquito, suppose a mosquito infected with second stage larva bites a new human host, it cannot infect the human host because the stages, or you can say microfilaria, are not present in their proboscis. Other indices include number of infective mosquitoes biting a human per month and how many mosquitoes are biting human beings per year. And finally, the infection risk per month and per year indicated by the number of infective larvae rather than the infective mosquitoes. So what did we learn today? Let us recapitulate. We learned that Wuchereria bancrofti is one of the most dreadful nematodes, largely confined to the tropical and subtropical regions of the world, affecting more than 120 million people. It is an endoparasite and is commonly found in the lymphatic vessels and lymph nodes of human beings, particularly in the growing regions, although it can be found in many other lymph nodes. Male and female Wuchereria are separate and exhibit distinct sexual dimorphism. Means male and female Wuchereria can be distinguished from various features. Wuchereria is a digenetic parasite and it requires two hosts to complete its life cycle. Man is the only primary host, while a large number of species of mosquitoes belonging to the genus Culex, Aedes and Anopheles act as secondary hosts. In India, Culex pipiens fatigans is the principal intermediate host, which is a nocturnal feeder, means it feeds only during night. Copulation between male and female adult takes place in the lymph glands of man. The female Wuchereria are ovoviviparous. After copulation, they liberate numerous Microfilariae in her lifetime. The Microfilariae are covered with the hyaline sheath and possess numerous nuclei which are absent from head, anal region, nerve ring and excretory pore of their body. These nuclei look like as granules. The Microfilariae enter the circulating blood of the human being from the lymphatic channels and do not undergo any further development in the human beings. Why? Because they need a lower temperature for their development. In India, Microfilariae exhibit nocturnal periodicity, as during night they appear in the peripheral blood vessels, especially between 10 p.m. to 4 a.m. to be sucked by Culex mosquitoes. However, during daytime, they reside in the large and deeper blood vessels of various organs of human body. When mosquitoes suck the blood of an infected individual, the Microfilariae covered by a sheath are ingested along with the blood and reach the stomach of Culex mosquito. Within 1 to 2 hours, they lose their sheath and they penetrate the wall of stomach. Then they migrate to the thoracic muscles or wing muscles of the mosquitoes for further development. Within next 14 days, the Microfilariae pass through 3 different larval stages. First larval stage, second and third. Third larval stage being the only infective stage, which then migrates to the proboscis of mosquito. Infection to a new host, or you can say human host, takes place only when the infected mosquito bites a human being. During biting, the larvae are deposited on the human skin from where they enter human body, generally through the site of mosquito bites. This is the most characteristic feature of Wuchereria, that they are not released into the human blood directly. Now the larvae accumulate in the lymph glands and they attain sexual maturity within 5 to 18 months. The intensity of filarial worm transmission depends upon various factors such as the number of infectious hosts carrying Microfilariae, biting rate of the Culex mosquitoes, the proportion of surviving mosquitoes which inhabit L3, or you can say third stage larvae. There are different indices also which affect the transmission of Wuchereria bancrofti. For example, how many times a mosquito bites a human being during a month, during a year, longevity of the mosquitoes, etc. I hope you will now be able to recognize Wuchereria morphologically and have understood its life cycle and transmission in human beings. This is a very important parasite. You must know about it. We will discuss the pathogenicity, diagnosis, prophylaxis, and prevention of this parasite in the next module. Thanks all of you for listening. Have a great day.